Good afternoon, class. Today we're going to be talking about muscles and muscle tissues. So, we're going to start off by talking about the three different types of muscle tissue: skeletal muscle, cardiac muscle, smooth muscle. And then we're going to go into more detail about the structure and function of muscle tissue in particular. An overview of muscle tissue. So, nearly half of the body mass is muscle tissue. Muscles can transform chemical energy, ATP, into direct mechanical energy, which is capable of exerting force. So, we are going to look at the types of muscle tissue. Again, cardiac, smooth, and skeletal, characteristics of muscle tissue, and the functions of muscles. All right, so when we talk about muscle tissues, we need to understand some terminology. Whenever you see the prefix myo, think of it's a root word meaning muscle, and then when you see the prefix sarco, okay, sarco is a has a both are has a root word meaning flesh, okay, and essentially with the terminology for muscle, so we already talked about the different organelles of a cell. So with muscle, instead of having saying cytoplasm, you just say sarcoplasm. You just put the sarco prefix in front of plasm. Okay, so sarcoplasm is the cytoplasm of muscle, and instead of calling a muscle cell plasma membrane, we say that muscle cells have a sarcolemma. Okay, so sarcolemma is what we say muscle cells have instead of a plasma membrane, sarcolemma. All right. So again, just understand that terminology, and then again, there are three types of muscle cells: skeletal, cardiac, and smooth. And skeletal and smooth muscle cells are elongated and are referred to as muscle fibers. So again, the skeletal and smooth cells are very long, and they're referred to as muscle fibers. All right, so when you talk about skeletal muscle, all right, so if you can, a lot of times it's easier to visualize things than to just write it. So if you can do like a brace map which shows the whole to part relationships, or you can just draw it out. But essentially, skeletal muscle is made of muscle fibers, okay? And these muscle fibers consist of a complex organelle called myofibrils. So again, a muscle fiber is an elongated, multinucleated cell. Okay, so muscle fiber is a cell, an elongated cell, and this elongated cell, this muscle fiber, is packed with an organelle. Okay, a complex organelle called myofibrils. So it's packed with hundreds or of myofibrils. Okay, it's packed with them. Um, but these myofibrils, okay, are made up of myofilaments. Okay, so again, if you can do a drawing of it or a brace map, so you kind of visualize it. So you have muscle tissue, which is made of muscle fibers, and muscle fibers are elongated multinucleated cells, okay? And within these cells, remember cells have organelles, but muscle cells have a special organelle called the myofibril, okay? All right, and they're packed full of these myofibrils, okay? And The myofibril are a rod-like contractile elements that occupy most of the muscle cell volume, and they're composed of sarcomeres, which are the contract unit of the muscle fiber, essentially. And the myofibril is made up of thin and thick filaments, and we're going to talk more about that later on. And we will have a diagram where you can visualize this better, but it's good to write it out for yourself so you can see how the muscle tissue is composed. All right, so skeletal muscle is also voluntary. So it's the only out of the three types of muscle tissue that we have voluntary control of. So we can say in our mind, I want to pick something up, and your biceps will contract so that you can pick something up. So they contract rapidly, they tire easily, and they're powerful. So when you think of skeletal muscle, think of striated and voluntary. Cardiac muscle is only tissue is only found in the heart. All right, it is striated too, but it is involuntary. So your heart has pacemaker cells, okay, which are going to create an electrical current that causes your heart to contract, and it does it without you thinking about it. It's involuntary. But when you think about cardiac muscle, think about it's also striated, but it's involuntary. And I'll talk about the difference, how you can tell the difference between cardiac muscle and skeletal muscle because they're both striated, but cardiac muscle has intercalated discs, which you'll see when we show a figure comparing the three types of muscle tissue. All right, so smooth muscle tissue is found in the walls of hollow organs like your stomach, urinary bladder. It has no striations and it's involuntary. Also, like cardiac muscle is involuntary, so it cannot be controlled consciously. So, like when we eat something, we don't have to tell our stomach, our digestive tract, to pass the food along. Okay, it does it involuntary. All right, so this is a figure showing you the location of the three types of muscle tissue. So again, your skeletal muscles attached to bones or to skin, cardiac muscle found in the walls of the heart, and then the smooth muscle found in hollow organs, visceral organs. And these figures down here allowed you to compare the appearance of the three types of muscle. So again, skeletal muscle, they're made of these long, elongated muscle fibers, muscle cells. Okay, they're long, elongated, and they have multiple nuclei, and they have these striations. Cardiac muscle also has striations, but they have these intercalated discs. Okay, so these intercalated discs allow you to tell the difference between skeletal muscle and cardiac muscle. And then smooth muscle have no striations, okay, but they are also elongated muscle fiber cells, but they're long cells too, just like skeletal muscle are long cells. All right, so characteristics of muscle tissue. All muscles share four main characteristics. They are excitable, so they can receive and respond to a stimulus by changing its membrane potential. In the case of muscles, the stimulus is usually a chemical, and it's usually for skeletal muscle, it's going to be a neurotransmitter released by a nerve cell. They have to be able to contract, have to be able to shorten. When stimulated, they got to be able to stretch. They got to be able to extend themselves, and they need to be elastic so they can recoil back into shape once they have finished contracting. So the four important functions of muscles. So they produce movement. They're responsible for all the locomotion and manipulation. They maintain posture and body position, stabilize joints, and they can generate heat as they contract. So that's one way that your body can maintain homeostasis. If it's cold outside, um, you shiver, and that shiver allows is your muscles contracting to generate heat. Some additional functions: they protect organs, form valves, control pupil size, and cause goosebumps. Skeletal muscle is an organ made up of different tissues with three features. It has a nerve and blood supply, and it also has a connective tissue sheath that's going to connect the skeletal muscle to bone and attachments. Each muscle receives a nerve, artery, and vein. So again, skeletal muscle we can consciously control skeletal muscle. So it has nerves supplying every fiber to control the activity. And skeletal muscle can contract, and when they contract, they're gonna require huge amounts of oxygen and nutrients. So that's why you need the blood supply, and you also need to get rid of waste products. So let's talk about now the connective tissue sheaths. So in an intact muscle, there are several different connective tissue sheaths. Together, these sheaths support each cell and reinforce and hold together the muscle, preventing the bulging muscles from bursting during exceptionally strong contractions. So let's talk about these connective tissue sheaths from external to internal. Each skeletal muscle, as well as each muscle fiber, is covered in connective tissue. So let's consider these connective tissue sheaths from external to internally. So the entire muscle is covered in the epimysium. So you can think of the epimysium as an overcoat of dense, irregular connective tissue that surrounds the whole muscle. All right, so the epimysium again is the overcoat. It's going to surround the entire muscle. Now remember that a muscle is composed of muscle cells, okay? And we call muscle cells fibers because the muscle cells are so elongated. All right, so that's why we call them muscle fibers because the muscle cells are so elongated. But these muscle fibers are grouped into bundles called fascicles, okay? And these fascicles are surrounded by a layer of dense, irregular connective tissue called perimysium. Perimysium. So the perimysium is going to surround each fascicle, and fascicles are bundles of muscle cells, bundles of muscle fibers. Now, each muscle fiber is also surrounded by its own connective tissue sheath, and each muscle fiber is surrounded by an endomysium. And the endomysium is a wispy sheath of connective tissue that surrounds each individual muscle fiber, and it consists of fine areolar connective tissue. Now, it's important to remember that all of these connective tissue sheaths are continuous with one another, as well as with the tendons that join muscles to bone. And when muscle fibers contract, they pull on these sheaths, which transmit the pulling force to the bone to be moved. This is a figure of what we got through talking about. So you can see right here, the epimysium surrounds the entire, so this connective tissue sheath surrounds the entire muscle, okay? So within a muscle, you have muscle cells, and we call these muscle cells fibers because they're long. So you have muscle fibers, okay? And you have bundles of muscle fibers which which are called fascicles. Okay, so you have bundles of muscle fibers which are called fascicles. So these are bundles of muscle fibers right here, and the fascicles are wrapped around by the perimysium. Okay, so you see right here, this is the perimysium surrounding a bundle of muscle fibers. All right, so now you have to remember that the muscle fiber is a cell, okay? It's an elongated cell, okay? So within the muscle fiber, within the muscle cell, you have a organelle, a complex organelle called myofibril. So within this muscle fiber right here, which is a muscle cell, you have all these little long sticks that you see. These are myofibrils, so they're organelles. These are organelles within the cell. So we talked about different organelles already with cells, Golgi, apparatus, mitochondria, and so forth. But these are a complex organelle within a muscle cell called myofibrils. So you have a bunch of myofibrils, all right, that compose a muscle that are within a muscle cell, okay, and each muscle fiber has its own connective tissue sheath, the endomysium. Okay, the endomysium surrounds the individual muscle fiber, and remember the individual muscle fiber is packed with the myofibril, which is an organelle, okay, the pack with these myofibrils. And again, it's important to remember that all of these connective tissue sheaths are continuous with one another, as well with the tendons that join muscles to bone. So all these connective tissue sheaths are continuous with each other, and you see right here this tendon which joins the muscle to bone. Okay. So this is a table that is summarizing what we just saw in the figure. So again, you have the epimysium that covers the entire muscle, okay? The epimysium. But muscle is composed of muscle cells, okay? But these muscle cells are going to be packed into bundles, okay? Packed into bundles. All right, and the bundles are going to be called fascicles, okay? So the muscle cells are packed into bundles called fascicles, and the fascicles are surrounded by the perimysium, okay? And remember that muscle cells are called muscle fibers because they're elongated, they're long, okay? And each muscle fiber, which is a muscle cell, has a complex organelle called myofibril. Okay, so it has hundreds of these myofibrils packed into the muscle cell. Okay, this organelle, the myofibril, packed into the muscle cell. But each muscle cell, each muscle fiber, is surrounded by its own connective tissue sheath called the endomysium. Okay, and again, all these connective tissue sheaths are continuous with one another. So now let's talk about muscles attachments. So again, muscles span joints and attach to bones, and they attach to bone in at least two places. The insertion, which is going to be the attachment to movable bone, and the origin is going to be the attachment to immovable or less movable bone. This is a figure showing you the insertion, which is the movable attachment to the movable bone, and it's showing you the origin. And generally, the origin doesn't move. Attachment to the immovable or slightly movable bone. Okay, and you always see that the insertion moves toward the origin. Okay, the insertion always moves toward the origin. So now we're going to go into more detail about the muscle fiber microanatomy. And again, muscle fibers are the muscle cells. They're just called fibers because they're elongated. All right. And within those cells, you have, they're packed with myofibrils, okay, which are a special type of organelle, okay, for contraction, okay. We'll talk about that in more detail in the next slides. But skeletal muscle fibers are long cells that contain multiple nuclei. The sarcolemma is the muscle fiber plasma membrane, and then the sarcoplasma is just the muscle fiber cytoplasm. And muscles because they take part in contraction. They need a lot of energy, so glycosomes, okay, so they supply glycogen. They store glycogen, which can be used as a glucose source for energy to generate ATP, and then you have myoglobin, which store oxygen. All right, so they need oxygen. So remember, in order to produce energy with aerobic respiration, right, you need oxygen for the electron transport chain. So um, again, in muscle cells need a lot of energy for contraction. All right, so within a muscle cell, within a muscle fiber, you have these modified organelles, the myofibrils, which consist of sarcomeres, which are the contract units of the muscle cell. And then you have the sarcoplasmic reticulum, which is just a type of smooth muscle. It's a smooth endoplasmic reticulum. So it's a smooth endoplasmic reticulum. That's just a type of smooth endoplasmic reticulum, and it stores calcium. And then you have T tubules, and T tubules are an extension of the plasma membrane, the muscle plasma membrane, extension of the sarcolemma. So the sarcolemma pushes down into what's called T tubules, and those T tubules are going to be important because when you are transmitting your action potential, your signal within a muscle cell, the T tubules make sure that all the muscle fibers are going to get excited. All right, so let's talk about in more detail each of these modified organelles. So again, each muscle cell, each muscle fiber is densely packed with a rod-like organelle called the myofibrils. Okay, so a single muscle fiber can contain thousands of these myofibrils, and they account for 80% of the muscle cell volume. Okay, so myofibrils are essentially made up of myofilaments. Okay, and these myofilaments are made of either actin or myosin. So if they're thin, they're made out of actin. If they're thick, they're made out of myosin. Okay, and essentially the myofibrils consist of these sarcomeres, and these sarcomeres are lined up end to end, and the sarcomeres are the contractile units of the muscle cell. Okay, and due to the um, thick and thin filament arrangement, you have these striations, all right, which are called dark and light bands. We're going to talk about those in more detail, but those are due to the arrangements of the myofilaments, the arrangements of the thick and thin filaments. All right, and we're going to talk about the molecular composition of myofilaments in more detail also. All right, so again, here's another figure of a muscle fiber. So remember, this is a muscle cell, and it's packed in its cytoplasm with all these myofibrils. Okay, and these myofibrils are made up of myofilaments. Okay, which are thick and thin filaments, and due to the arrangement of these thick and thin filaments, also called dark and light bands, the thick filaments are the dark bands, the thin filaments are the light bands, and due to the arrangements of the thick and thin filaments, you have these striations, dark and light bands. Okay, and this is the sarcolemma, okay, which is the plasma membrane of the muscle cell. And again, this sarcolemma is going to extend down into what's called T tubules, and you'll see a picture of that in a figure later on. So let's talk about the striations in muscle. So the striations are due to repeating series of dark and light bands. So the dark bands um, are going to be the A bands, okay? And essentially the A bands consist of, it's easier to see this when you have a figure. We will see a figure in slides ahead. But essentially the A band has it's mostly what has thick and thin filaments. Thick filaments with thin filaments overlapping, but it has in the middle of the A band, you have an H zone, okay? And this H zone doesn't have any thin filaments in that area. So the H zone essentially of the A band appears less dense because the thin filaments do not extend into this region. And within the H zone, within this H zone, you have an M line. Okay, this M line is the center of the H zone and is slightly darker because of the fine protein strands that hold adjacent thick filaments together. So this M line is going to be what holds adjacent thick filaments together. All right, and then you have these I bands. So these I bands are composed strictly of thin filaments. Okay, so they're just thin filaments, and they have Z discs. Okay, so each light I band has a midline interruption, a darker area called the Z disc. The thin filaments are held in alignment at the Z disc, and they are anchored to the sarcolemma at the Z disc also. So the Z disc anchors to the sarcolemma, which is the plasma membrane of the muscle cell. this is just a photograph of two isolated muscle fibers. So again, you can see the striations, the dark and light bands clearly, and you can see the nuclei. Okay. The region of a myofibril between two successive Z discs is a sarcomere. And a sarcomere is the smallest contractile unit of a muscle fiber, and it contains an A band flanked by half an I band at each end. And individual sarcomeres align end to end along myofibril like boxcars in a train. All right, so this is the a figure of a myofibril so that you can see the myofilaments. So we've already talked about the A band, which is the dark band. So on this figure, the thin filaments are blue and the thick filaments are red. And you can see right here, this is the sarcomere, and it consists of an A band flanked by half of an I band on both sides. And you also see in the A band, which is right here, you see that you have it has an entire thick filaments, okay, and then you have overlapping thin filaments. But right here in the middle of the A band, you have this H zone, which is bare of, there are no thin filaments in this H zone, okay, and in this H zone, you have the M line, okay? This M line is where the attaching point for the thick filaments, okay? At least that M line, all right? And then you see right here with your I band, your light bands, it's all thin filaments. It's all thin filaments, and you see your Z disc right there. The Z disc holds the thin filaments in alignment, and it is anchored to the sarcolemma. Okay, it's anchored to the sarcolemma, the Z disc. So again, the myofilaments or orderly arrangements of actin and myosin. And actin is going to be found in the thin filaments. Myosin is going to be found in the thick filaments. Okay? And the actin myofilaments or the thin filaments, and they extend, remember from my diagram that we just saw, all the way across the I band, and then they are, so the I band is nothing but thin filaments. Remember, the I band is nothing but thin filaments, and then the A band is an entire thick filament, but it has overlapping thin filaments. But it has that in the A band, you have that bare H zone, which has no thin filament. So that's why it's only partly in the A band. And then the myosin microfilament, again, it's the entire length of the A band. It's the thick filaments, and the myosin myofilaments are connected at the M line. All right, and I should have mentioned it with the thin filaments, they're anchored to the Z disc, and the Z disc also anchors the thin filaments to the sarcolemma, the plasma membrane. So we're going to see this on this next slide. The sarcomere cross-section shows a hexagonal arrangement of one thick filament surrounded by 6 thin filaments. And we are going to see that in this next slide. So this is showing you the arrangement of how you have the hexagonal arrangement of on this image right here of 6 thin filaments surrounded each thick filament, and 3 thick filaments enclose each thin filament. So that's what this is showing you right there. All right, so this is showing you, remember in the I band, you only see light blue little dots. So the I band only has thin filaments only. So at the H zone, remember the H zone is this middle part of the A band, okay? In the H zone, which is the middle part of the A band, you only have thick filaments, okay? And you also have the M line in the H zone, okay? And the M line is where the thick filaments are linked by accessory proteins. So this is what this is showing. The thick filaments are linked by accessory proteins. So that's what that's showing. And this is just showing you a sarcomere, all right, which is an I band with half of a I band. I'm sorry, it's an A band, entire A band with half of an I band on each side, flanked by half an part of an I band on each side. Okay, an A band flanked by half of an I band on each side. That's a sarcomere. And you can also see on the thick filaments, you see these myosin heads, and these blue ones are the thin filaments. So essentially, when a muscle contracts, these myosin heads are going to bind to the thin filaments, okay, and then it's going to use ATP. It's going to cleave ATP, and it's going to move, causing the thin filament to slide past it. And essentially what's going to happen is the thin filament is going to slide past the thick filament, and this H zone, which normally has no thick filaments, is going to disappear because the thin filaments are going to slide into that area, okay, past the thick filaments. It's going to slide into that H zone area. So that H zone it's going to disappear. It's no longer going to be just thick filaments. And just to summarize this again, when a muscle contracts, again, these myosin heads are going to bind the thin filaments, okay, and then the I band is going to slide toward the center, toward the center of the H zone, and the H zone is going to disappear, okay? So, and it's also important to know that the A band move closer together, but their actual length doesn't actually change. They're just moving. You see how this is an A band, right? Well, light band. I should say this right here. Light band right here. Light band right here. But the A band move closer together, but the length doesn't actually change. Okay, so these thin filaments are going to move toward each other in the center right here. Okay. So let's talk about the composition of myofilaments. So with the thick filaments, remember they have the protein myosin, all right, that contains 2 heavy and 4 light polypeptide chains. So during contraction, the heads are going to link to the thin filaments and form cross bridges. And the thin filaments are composed of actin, all right? So let's look at an image of that. So you see right here the thick filaments, you see these myosin heads, okay? And remember, when these myosin heads bind and then slides and then moves, causes the thin filaments to slide past it, it's going to need ATP. So these are the ATP binding sites, okay? And this is the actin binding site where it's going to bind to on the thin filaments, all right? So that's the head region, and this is the tail region. And this is showing you the thin filament. So it consists of 2 strands of actin subunits twisted into a helix plus 2 types of regulatory proteins, all right, troponin and tropomyosin. So these are the active sites for myosin attachments right here. All right, so you see that on the actin subunits. And this is just a look like a scanning electron microscope image of thin and thick filaments, and you can see the myosin heads. And this is just showing you again, in an A band, you have an entire thick filament. So you have entire thick filament, and then you have these overlapping thin filaments, okay? And then you have this H zone, which is the bare zone, where it has no thin filaments, okay? But then when a contraction occurs, these myosin heads are going to bind the thin filaments, and it's going to cause the thin filaments to slide toward the center, okay? The thin filaments are going to slide toward each other, toward the center, and it's going to cause that H zone, this H zone, to disappear when these thin filaments slide toward the center. The sarcoplasmic reticulum is an elaborate, smooth endoplasmic reticulum. The sarcoplasmic reticulum regulates intracellular levels of calcium. So it stores calcium and releases it on demand when a muscle fiber is stimulated to contract. And interconnecting tubules of sarcoplasmic reticulum surround each myofibril, the way that a sleeve of a loosely knitted sweater surrounds your arm. And most sarcoplasmic reticulum tubules run along the myofibril, communicating with each other at the H zone. You also have terminal cisterns, which are just end sacs. So the sarcoplasmic reticulum has these terminal cisterns, which are end sacs, which form larger perpendicular cross channels at the A band, I band junctions. And when at these AI band junctions, you have these 2 T tubules, which we'll talk about in a second, in the middle. So you have the T tubules in the middle, kind of sandwiched in between 2 terminal cisterns of the sarcoplasmic reticulum, and they form, that's what those are called, a triad. Okay, and the tubules are just protrusions of the sarcolemma, which is the plasma membrane of the muscle cell, of the muscle fiber. So it's just protrusions of the sarcolemma deep into the cell interior. All right, so it increases the muscle fiber surface area greatly, and it allows for the electrical nerve signal to reach deep into the interior of each muscle fiber. So let's talk a little bit more about the triad relationship. So the roles of the T tubules and the sarcoplasmic reticulum in providing signals for contraction are tightly linked. At the triads, you have membrane spanning proteins from the T tubules and sarcoplasmic reticulum linked together across the gap between the two membranes, and the protruding integral proteins of the T tubule act as a voltage sensor, and then the integral proteins of the sarcoplasmic reticulum form gated channels through which the terminal cisterns release calcium. So essentially, the T tubules detect the voltage change, and then that voltage change is going to open up the voltage gated channels of the sarcoplasmic reticulum, which are going to release calcium through the terminal cisterns. So this figure just shows what we got through talking about. So again, you have your sarcoplasmic reticulum, which is smooth endoplasmic reticulum. You see how it wraps around the myofibril, kind of like a sweater wraps around your arm. Okay, and then in the middle at the H zone, the sarcoplasmic reticulum communicate with each other, but at the A band, I band junction, you have these terminal cisterns, which just are in sacs of these sarcoplasm reticulum. Okay, and then right here you have the T tubule, is the lights, real light blue, or the T tubule, and you see how the terminal cisterns, the end sacs of the sarcoplasmic reticulum, terminal cisterns sandwich the T tubule. Okay, that's called a triad. Okay, and the T tubule is just an extension of the sarcolemma. Remember, the sarcolemma is the plasma membrane of the muscle fiber. So the T cell is just a protrusion of the sarcolemma, and it has a voltage sensor. And when you have a action potential when it's going through a cell, essentially you're going to have electronic depolarization, and that's going to send a signal that can be detected by the T tubules. And then you have at these triads, you have the terminal cisterns, which have voltage gated calcium channels, are going to be open. Those voltage gated calcium channels in the terminal systems are going to open and release calcium. All right. All right. So the sliding filament model of contraction in the relaxed state, the thick and thin filaments overlap only slightly at ends of the A band. And remember, in the A band, in the H zone, it has only thick filaments. There's no thin filaments in the A band. That's that H zone. The sliding filament model of contraction states that during contraction, thin filaments slide past thick filaments, causing actin and myosin to overlap more. Neither thick nor thin filaments change length, just overlap more. When the nervous system stimulates muscle fiber, myosin heads are allowed to bind to actin, forming cross bridges, which causes sliding process to begin. Cross bridge attachments form and break several times, each time pulling thin filaments a little bit closer toward the center of a sarcomere in a ratcheting action, causing shortening of the muscle fiber. So the Z discs are pulled toward the M line, the I bands shorten, and the Z discs become closer. In the H zone, it's going to disappear, and the A bands move closer to each other. And so this is showing you a relaxed sarcomere muscle fiber. You see you have this H zone, has only the thick filaments, okay? And the myosin heads are not connected. There's no cross bridge, and it's not connected to the thin filaments. All right, and you see this H zone is present. Only thick filaments are there. All right, so this is a contracted sarcomere. You see Your myosin heads are bound to the actin on the thin filaments, and they form these cross bridging. And you see how they have pulled that it's resulting in the thin filaments to be pulled centrally, all right, toward each other, and then the H zone essentially disappears because now you have the thin filaments are now present in the H zone, so the H zone disappears. All right, we will stop here for this lecture, and then the next lecture we will pick up talking about action potentials, how skeletal muscles contract.